Hey guys, in this video we're going to learn some important definitions. I'm going to start sharing my screen with you now, and you'll see our slides, which are under chapter 10 on Blackboard. So in this video we're going to learn what's a codon, what's an anti-codon, what are the start and stop codons, and then we're going to also talk about or revise the structure of the ribosome. So when we do our next part of protein synthesis, we're going to do a process called translation. So if you, again, thought of the English language term for this idea of translation, you would be converting your one language to another language, right? So here, we've got a translator, a machine that can translate one language to another. So our ribosome is our translator inside our cell. It's able to convert DNA language, which is A's and T's and G's and C's, right? And it's able to convert that into the protein language, which are going to be the amino acids, putting the right amino acids in the right order. So the ribosome is where translation is going to take place, and ribosomes are found in your cytoplasm. Remember, sometimes there are free ones just floating in the cytoplasm. Other times they're attached to the rough endoplasmic reticulum. But either way, our messenger RNA is going to have to come out of the nucleus through the nuclear envelope pores and go to the ribosome in the cytoplasm. And then the ribosome is going to translate the DNA language and the RNA language, the nucleotide language, into protein language, amino acids, the right amino acids in the right order. So there's something we're going to use for our problems that are going to be on our exams, and it's called the codon dictionary, right? So if you're learning another language, you usually have a English to French dictionary or a French to English dictionary where you can look up the word. So in our codon dictionary that we're going to use, you will see the trio of nucleotides that code for a specific amino acid. So if you see the three nucleotides UUU, then that codes for the amino acid phenylalanine. So this trio of nucleotides that codes for a specific amino acid is called a codon. So a codon is three nucleotides together. So you can see here, if you have C, A, and U together, that codes for histidine. You can see if you got C, C, and U, that codes for proline. If you have CCC, that codes for proline. So you can see that there are certain trios of nucleotides that code for specific amino acids. And you'll notice proline, 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 proline. Proline here has 1, 2, 3, 4 codons, and they all code for the same amino acid, proline. And we'll talk about why we need that later when we learn about mutations. But for now, you can see how this is going to work. If you know that your amino acid has a specific trio of, or if you know that your messenger RNA has a specific trio of nucleotides. So if your messenger RNA says CGU, right? You're going to look this up in your codon dictionary. So you'll notice the first letter C, you get your first base here. So here's everything that starts with the C. Your second base is G, right? Find G. So then across C, down G, and third base is U. CGU codes for arginine. So you would know if your messenger RNA has the trio CGU, you would put in the amino acid arginine in that position on your protein. So because we have the 20 amino acids that we learned about, or 21 amino acids that we learned about, there are more codons than amino acids. So there are several combinations of nucleotides that could lead you to put in a specific amino acid, as we saw here with the proline. Now, there are some very special codons. There is the start codon, which is AUG. So when you see AUG on your messenger RNA, that's where you start building your protein. And the first amino acid that always goes in every polypeptide is going to be methionine, because that's the amino acid that the start codon codes for. So AUG, this is your signal that you should start adding amino acids and building your protein here. Then there's 3 stop codons, UAA, UAG, UGA. And you'll notice that these 3 stop codons don't have an amino acid name underneath them. they don't code for an amino acid. So when the ribosome sees the stop codon, there's no amino acid that it can put into that area, so it just stops making the protein and breaks apart. So the stop codon tells you where to stop building your protein. Your start codon tells you where to start building your protein. So like I said, most amino acids have more than one codon, and this protects you against mutations. If you have a small mistake, one base mistake in your DNA, you might still be able to make the correct protein because we've got this built-in idea of there are more than one codon for each amino acid, right? So most amino acids have more than one codon, but there's only one start codon, it's AUG. There are 3 stop codons, UAA, UGA, UAG. And the interesting thing about this codon dictionary is it's universal. If you look in an amoeba, if you look in a bacteria like Staphylococcus aureus, if you look in many organisms across many areas of the world, every organism uses this same codon dictionary. So this is another really important idea, that maybe we all evolved along the same pathways, or maybe we all evolved from these cells that were there at the start of the earth, right? Or at the start of life on Earth. So the fact that the code is universal, that it's found in amoebas, bacteria, fungi, every living thing uses the same code. So UCU would code for serine in a bacteria, in an amoeba, in a fungus, in a human, in a dog. The codon dictionary is the same for all the organisms. And this is a big clue that we must have all evolved from each other, and that's how we have this thing in common. So another important thing that we're going to need to build our protein is this structure, which is called a transfer RNA, or tRNA. So the job of the transfer RNA is to bring the amino acid to the ribosome. Remember, the ribosome is going to assemble the amino acids in the right order, but somebody has to be delivering the amino acids to the ribosome so that it gets the right amino acid at the right time. So that's where this transfer RNA comes in. And you can see the structure of the transfer RNA. It kind of looks like a cross. On one end, you've got your amino acid attached to one end of your transfer RNA, and at the other end, you have this very specific structure called the anti-codon. So we know that on our messenger RNA, we have a codon, a trio of nucleotide bases that codes for a specific amino acid. So you can see here that the anti-codon is on the transfer RNA, and the anti-codon is a complementary match to the codon. So when we're trying to bring in the right amino acid at the right time, the codon is read by the ribosome. It gets the transfer RNA with the matching anti-codon, and then that brings the right amino acid into place. And this will make a lot more sense when you see the video of how translation works. But the idea here is that the transfer RNA is going to collect the correct amino acid from the pool of amino acids that are in our cytoplasm. Just like we had a pool of bases, A's and T's and G's and C's in our nucleus, we have a pool of amino acids in the cytoplasm of our cell. So our transfer RNA grabs the right amino acid and brings it to the ribosome so that we can get the right amino acid at the right time to build our protein. So the two important parts of our transfer RNA is that one end we've got the anti-codon, and the other end we've got our amino acid. So on this side, here's our amino acid attached to our transfer RNA. And then down the other side, we have the anti-codon. And remember, the anti-codon is going to be complementary to the codon. So the anti-codon is going to complement the codon. So see here we had CUU, and the anti-codon is GAA, right? So the G matches the C, the A matches the U, the A matches the U. So the anti-codon is the complementary match of the codon. So the transfer RNA helps to interpret and to communicate and to change or translate the language of your nucleotides down here into the language of your protein up here. And when you see how this works in the next video, it's going to make a lot more sense to you. Now, this shows you what a ribosome looks like. And remember, ribosomes are made of ribosomal RNA or RNA and protein. So the ribosome structure is just made of ribosomal RNA. Remember, ribosomal RNA is made in your nucleolus. So our ribosome is made of two parts, the large subunit and the small subunit, right? So this part down here below the messenger RNA, this section, that's your small subunit. This is your large subunit. So it's two pieces that come together and they kind of sandwich on top of your messenger RNA. So see this pink thing in the center? This is your messenger RNA. Your small subunit binds the bottom of the messenger RNA and the large subunit sits on top of the messenger RNA. So the messenger RNA is kind of sandwiched between these two subunits. And you can see that the ribosome has 3 sites inside the large subunit. There's an A site, a P site, and an E site. So let's see what these stand for. The A site is where the amino acid comes in. The P site is where the amino acids are stuck together, so it's called the peptide site. And then the E site is the exit. This is where your empty transfer RNA is going to leave your ribosome. So I like to think the amino acid is accepted at the A site. It's stuck together and formed a peptide at the P site, and then E for exit, right? This is where the empty transfer RNA, tRNA, is going to exit the ribosome. So it's coming in, it's sliding over, it's exiting, it's off, right? So we need to know the names of the 3 sites. The A is for where the amino acid approaches or comes in. The P is where the amino acids stick together to form a peptide, and the E is where the transfer RNA exits after it's dropped off the amino acid. So like I said, a lot of this stuff is going to make much more sense when you actually see the next videos and how translation actually works. So stick with it, because when you see how it all comes together, it'll make a lot more sense to you.